going to continue our discussion of Mendelian genetics or classical genetics, actually looking at problems today and seeing how to solve problems, figure out who has what genotypes, and based on what phenotypes are being expressed. So anyway, where we want to begin with this, though, is to review quickly the 6 basic crosses that we've discussed and the ratios generated by those. So again, if you cross a homozygous dominant with a homozygous dominant, there's only one possible outcome, 100% homozygous dominant. And if we're looking at the purple versus white flowers that we discussed last time, then 100% of the offspring would have to be purple. And we're also, again, working with complete dominance. Okay, again, if you cross homozygous recessive with homozygous recessive, only possible outcome here would be 100% homozygous recessive times homozygous recessive. The only possible outcome would be 100% recessive. All the offspring would have white flowers in this situation. If you cross homozygous dominant with homozygous recessive, we would have 100% heterozygous outcome, 100% purple flowers. If you cross heterozygotes, you would end up with 25% homozygous dominant, half heterozygous, 25% or a quarter homozygous recessive, which would mean 3/4 purple or 75% purple, 1/4 white. And then homozygous dominant times heterozygous would give us half homozygous dominant, half heterozygous, 100% purple. And homozygous recessive times heterozygous would give us, and actually it isn't showing us that, but it would give us half heterozygous and half homozygous recessive. And also half would be purple and half would be white. So again, this is all review from our last lesson. Also, again, unless I tell you otherwise, we're going to assume complete dominance. And by that I mean then that if we're looking at homozygous dominant and heterozygous, that they will have the same phenotype. So in this case, homozygous dominant and heterozygous would both express purple flowers. And you can't tell by looking at the flower whether they're homozygous dominant or heterozygous. And in this case, homozygous recessive would show us white. So again, the only way to show recessive phenotypes is to be homozygous recessive when we're talking about complete dominance. Now, there is another type of dominance, which is called incomplete dominance. And so I want to quickly show you that. We won't be doing many problems with this, but when you see it, it's very convenient to work with. Now, with incomplete dominance, again, we're going to work with flower color, but this would be a different kind of flower. This is what's known as the 4 o'clock. And in the 4 o'clock plant, there's some different pigments potentially here. So one would be red versus white. So red is dominant over white. And so again, we could have our 3 gene pairs, homozygous dominant, heterozygous, homozygous recessive. But in incomplete dominance, what happens is that if you're homozygous dominant, you will show one extreme of the phenotype. So in this case, red flowers. But the heterozygous will be incompletely dominant such that it will be a lighter shade. So this would be pink. So pink flowers would be heterozygous. And then homozygous recessive would be white, or is white in this particular case. So this goes back to that blending idea. So one of the things that's kind of interesting about Mendel's work is that Mendel picked 6 different traits to look at. They all happen to have complete dominance. But there are situations in nature where there is this incomplete dominance, so that we actually have 3 different phenotypes instead of just 2 phenotypes. Now, what's nice about this is that now phenotype tells you genotype. So again, if you have red, that would be homozygous dominant. Pink flowers with the 4 o'clock would be heterozygous. White flowers would be homozygous recessive. So when you're working with incomplete dominance, it's actually really easy to work with because anytime you see a phenotype, it tells you what the genotype is. So again, if you were crossing red flowers with white flowers in this situation, the red flowers would only be able to pass on dominant alleles. The white flowers would only be able to pass on recessive alleles. So if any of these gametes came together with any of these gametes, it would produce heterozygotes, all of which are pink. And then if you crossed that F1 generation, you'd end up with 1/4 red, which would be homozygous dominance, half pink, which are heterozygotes, and 1/4 white, which would be homozygous recessives. So again, pretty convenient because the phenotype here, physical appearance, tells you the genotype. So again, that's incomplete dominance. So for our purposes and our problems, however, we're going to work primarily with complete dominance, where there is no difference between the homozygous dominant and the heterozygote. So assume complete dominance unless there's some reason not to assume that. And so again, as I just mentioned, so quarter would be red, half pink, quarter white. Okay, now we're going to go out, now I want to look at a few examples of what are known as pedigrees. And so in pedigrees, essentially what the key is that if we see squares, that represents a male, a circle will represent females, and then usually you're interested in one trait or another. So an affected trait or a trait of interest to you will be shaded in some way. So it may be black, it may be gray, it depends on whatever the author wants to use. So again, we'll be tracking some physical trait, some phenotype, and so either the male or females will be shaded. If individuals mate, a line is drawn between them, and then their offspring will also be linked together with a line, and then a line connects those two as well. So we'll see one generation and then the next generation. And so what we want to do then is start with phenotypes and build to genotypes, try to figure out what all the gene pairs are for individuals. All right, so let's look at an example. So one example in humans is widow's peak. And so the widow's peak is this kind of V-shaped hairline here as opposed to a smooth hairline here. And as it turns out, widow's peak in humans is dominant over a straight hairline or smooth hairline. So again, when you look at someone, and we're going to assume complete dominance. So if someone shows a widow's peak, then they could be homozygous dominant or heterozygous. If someone does not show a widow's peak, they have to be homozygous recessive. So again, that's what defines recessive traits. Recessive traits can be hidden. And so the only way recessive phenotypes show up is if an individual is homozygous recessive. Now, when we look at the pedigree then, we're looking at males and females here. So parents and offspring, parents and offspring. And then these mated and produced offspring here as well. Now, we're interested in the shaded trait. And in this case, the shaded trait is showing us those individuals that have a widow's peak. Now, by looking at a pedigree, you can sometimes infer whether a trait is whether a phenotype is encoded by dominant alleles or recessive alleles. And you can do that with recessive phenotypes for sure. It's harder with dominant phenotypes. But if we look at this, in each generation we see the shaded trait. We see it in this generation, we see it in this generation, we see it in this generation, we see it here, we see it here. So in other words, the trait is not being hidden in any of the generations. So we couldn't be positive that it's carried by a dominant allele, but it's a pretty good indicator that it's carried by a dominant allele. And I'll show you how this contrasts with recessive phenotypes and recessive alleles a little bit later. All right, so again, those that are shaded then have widow's peaks. Those that are not shaded do not have widow's peak. So if we were trying to figure out all the possible genotypes that we can, which one tells us, which phenotype tells us genotype? Hopefully you're catching on that if you show a recessive phenotype, you have to be homozygous recessive. So we always want to start with what we know. And what we know here is the genotypes of all the individuals that lack a widow's peak. They have to be homozygous recessive. So this individual is homozygous recessive, this one, and so on down the line. These are all homozygous recessive because they show the recessive phenotype. If they only had one recessive allele and one dominant allele, in other words, if they were heterozygous, the dominant allele would cover up and hide the recessive allele, they would have a widow's peak. So the size of the widow peak has nothing to do with whether you're homozygous dominant or recessive. That's either you have one or you don't. Okay, now once you get that down, and again, what I would encourage you, I'm showing you how to do this first pedigree, but you should certainly, I think, copy these down or use your lecture outline and try to do them yourself before I solve them for you to see if you can figure these out. But I'm going to go ahead and continue on now, but certainly, again, you need to put pencil to paper and not just try to figure this stuff out in your head, but actually, write it down and try to figure it out. Now, keep in mind, principle of segregation, you inherit two alleles, you only get to pass on one allele to your offspring, to a particular offspring, and then your mate's going to pass on the other allele. So, as we move along here, again, these individuals only have recessive alleles to pass on. So, if we look at these individuals here, both homozygous recessive, one recessive allele came from this parent, that means one recessive allele had to come from this parent. So this parent, even though they have a widow's peak, they have a recessive allele. That's the only way they could produce homozygous recessive offspring. Okay? So one recessive allele from this parent, one recessive allele from this parent, but they show the dominant phenotype, so they have to be heterozygous. So again, it is possible to hide recessive alleles. Okay, let's look at another individual. This individual here has to be heterozygous. Why? Because the parent only has recessive alleles to pass on. So this parent only has recessive alleles to pass on, yet they show the dominant phenotype, so that dominant allele had to come from here. If we continue on, this individual has to be heterozygous as well, again, because this same parent only has recessive alleles to pass on, yet they show the dominant phenotype, so they have to be heterozygous. Continuing on, this individual has to be heterozygous because the daughter is homozygous recessive. So again, one recessive allele came from dad, one recessive allele came from mom. So this mom has to be heterozygous. And then last but not least, then we only have really this genotype to figure out. All the others we can be certain of. Oh, I take that back. This daughter also then would have to be heterozygous because this parent only has a recessive allele to pass on. So these two parents then are heterozygous. And so as we look at this individual, this daughter, we really can't be certain whether she's homozygous dominant or heterozygous. If she had a daughter that lacked a widow's peak, then we would know she was heterozygous. But that, with what information we have right now, she could be homozygous dominant, she could be heterozygous, we can't be sure. But the rest of these we can all be sure of by just following the concept, you inherit two alleles, pass on one. If you only have recessive alleles to pass on, that's all you can pass on. And you can only pass on one of those. So that means if there are two recessive alleles, one had to come from the other parent as well. So again, with these pedigrees, you want to start with what you know, basically start with the homozygous recessives, and then just follow alleles up and down until you figure out all the possible genotypes. Okay, so let's look at another pedigree. This is a pedigree involving earlobes. Attached earlobes, that phenotype is recessive to having a free earlobe. So if you have a free earlobe, you could be homozygous dominant or heterozygous. Now, in this pedigree, the shaded individuals have attached earlobes. Now, we could, even if I hadn't told you this already, you can see that this is dominant and this is recessive. But if I hadn't told you that, you could figure that out just by looking at the pedigree. In other words, just looking at the shaded trait, you could figure out whether it's encoded by dominant or recessive alleles. And the clue really is in this side of the pedigree right here. The parents don't show the trait, but the offspring do. The only way you can hide a trait is if that trait is encoded by recessive alleles. So even though I told you this, if I hadn't told you this, that's the clue. Offspring show the trait, parents don't. So that trait's encoded by recessive alleles. So the attached earlobes are the shaded trait here, so attached earlobes are recessive to dominant earlobes. So again, the idea is that if you have free earlobes. So if you have free earlobes then, that's the dominant trait. You could be homozygous dominant, you could be heterozygous. And I'm just using the letter E now to represent the gene E, gene for ears rather. And then if you're homozygous recessive, that's the only way you can show the recessive phenotype, attached earlobes. So start with what we know. Everybody who has attached earlobes has to be homozygous recessive. So let's go ahead and get that in. And the letters don't matter. You could pick any letter you want, but we're using lowercase to represent recessive alleles. Okay, then it's just a matter of starting to work up and down, if you will, to figure out what the other genotypes are. So again, we're seeing two homozygous recessives here. So each of these parents has to have a recessive allele. We're seeing homozygous recessive here, so this parent has to have a recessive allele. We're seeing homozygous recessive here. This parent and this parent have to each have a recessive allele. So again, let's go ahead and build our pedigree based on that kind of information. So again, this parent has to be heterozygous because one recessive allele from this parent, one recessive allele from this parent. That parent does not have free earlobes, attached earlobes, has free earlobes, so that one has to be heterozygous. So again, heterozygous parents are hiding the attached earlobes, but you do have offspring with the attached earlobes. Continuing on. Again, this offspring here could be either, because we would need more information to figure that out. So we just know that there are free earlobes, could be homozygous dominant, could be heterozygous, because if you cross this in a Punnett square, those are both possible outcomes there. This parent has to be heterozygous, again, because homozygous recessive, one recessive allele from here, one recessive allele from here. It has to be heterozygous because The daughter is homozygous recessive, so one recessive allele from this dad, one recessive allele from mom. So that makes her heterozygous as well. And then, last but not least, this daughter could be either. So we know she has free earlobes, could be homozygous dominant, could be heterozygous. And again, if you do your Punnett square, you'll realize those are each possible crosses when you cross heterozygotes. Okay, let's look at another pedigree. And again, it doesn't really even matter what it is. It doesn't even matter what the trait is for. What we're looking at is a shaded trait for some physical characteristic. And what we're trying to figure out, first of all, is this shaded characteristic encoded by dominant alleles or recessive alleles? So again, it might be a good idea to stop the module here and see if you can figure that out. The answer is that this shaded trait is encoded by recessive alleles. And the reason I know that is these two parents do not show the shaded trait. And again, I don't even care what the shaded trait is. They do not show the shaded trait, but their child does. So that trait was being hidden by the parents, and it shows up in the child. So that has to be encoded by a recessive allele. So I'm just arbitrarily picking the letter A to represent the dominant phenotype here and lowercase a to represent the recessive phenotype. So the shaded trait then, all of these individuals are showing the recessive phenotypes, so I'll just go ahead and label them as homozygous recessive. They have to be homozygous recessive to show the recessive phenotypes. And then again, we follow alleles, either down to the next generation or from the previous generation. So again, if we're looking at these individuals, both homozygous recessive, one recessive allele from here, one recessive allele from dad. She has two recessive alleles, one had to come from either parent here. So we'll just go ahead and continue to build as many genotypes as possible. So again, homozygous recessives tell us that this parent has to be heterozygous. This son has to be heterozygous because mom only has recessive alleles to pass on, yet he shows the dominant phenotype. Anne has to be heterozygous because mom only has recessive alleles to pass on, yet she shows the dominant phenotype. Michael has to be heterozygous because homozygous recessive child, one recessive allele from dad, one recessive allele, or from mom rather, one recessive allele from dad, so it has to be heterozygous. And then here, this son has to be heterozygous because the dad only has recessive alleles to pass on. And again, this son has to be heterozygous because dad only has recessive alleles to pass on. It shows the dominant phenotype. This daughter, or excuse me, this woman we can't be sure of because neither child was homozygous recessive. So it could be homozygous dominant, could be heterozygous, can't be sure of that. Can't be sure of this parent either, Tina. Just know that she shows the dominant phenotype. And as a result, can't be positive of this son here either. Just know that he shows the dominant phenotype. So again, they're kind of fun to do in a way, and you can actually do some for your own family when you look at certain traits. Now, let's look at a slightly more complex example now. Now, every gene that we've talked about to this point, we've only had two alleles, either a dominant allele or a single recessive allele. And we've only looked at one allele being a dominant allele to a single recessive allele. Now, there are other situations that can occur out there. One is that there can be more than two alleles, so-called multiple allele inheritance. So there may be more than two versions of genes at a particular place in a chromosome, at a particular locus. And so that complicates matters. So there's going to be more than just two or three phenotypes as a result. Also, if you have multiple alleles, there may be what's known as codominance. More than one of those may be dominant. So one of the examples that we see in this is in what's known as the Landsteiner blood types. So when we look at Landsteiner blood types, you can be type A, type B, type AB, or O. And the alleles that are involved here is that there's an allele A that codes for a certain protein A. So if you have type A blood, your red blood cells then have this protein A embedded in the plasma membranes. There's an allele B, which is also a dominant allele. Both A and B are dominants. If you inherit allele B, you produce a protein B, a different kind of protein, different shape. And so your red blood cells then will express that protein. And then the allele I is the recessive allele here. And so the allele I does not code for either the A or B protein. So if we look at genotypes and phenotypes then, you could be homozygous for A. so-called AA. Or you could be AI. In either case, the A allele is a dominant allele, and so your red blood cells will show the A protein. You could be genotype BB or BI, in which case you will express the B protein in your red blood cells. And again, you'd be type B here as opposed to type A. What if you inherited an A allele and a B allele? They're both dominant, so both proteins would be made, and so your red blood cells would show both A and B proteins, meaning that you're type AB blood. And then last but not least, if you're homozygous for the I, if you're homozygous recessive, you're not producing either protein, and so that defines you as type O. So let's go ahead and look at a problem then showing codominance with these multiple alleles in the Landsteiner blood groupings. So again, Landsteiner blood groups show both codominance and multiple alleles at a particular locus for a particular gene. All right, so let's work off a pedigree again. So again, we have males and females, individual of type A blood, type B blood, type A blood, type A blood, and 2 O's. And let's try and figure out as many genotypes as we possibly can. Now again, you always start with what you know. And I want to go back to the previous slide because again, we want to look at genotypes and phenotypes. And in this particular case, again, if you're type A blood, you could be AA or AI. So even though you're type A, we don't know for sure your genotype. If you're type B, we don't know for sure your genotype. But if you're type AB, we know that you are genotype AB, and if you're type O, we know that you're a genotype II. So let me move ahead then to that pedigree again. And again, starting with what we know. We can't be positive of the genotypes here, but we can be positive of these two genotypes. So if a person is type O, they have to be type II. Okay, so that would be the genotype there. So let's go ahead and get those down. And this is actually a pretty tough problem. Some of these this is one of the tougher problems that we'll be doing. Now, given that, where can we go with the recessive alleles, and then what can we determine from that? Well, because this parent is homozygous for I, she only has I alleles to pass on to her son. So he's type A, but he has to be AI. He can't be AA because she does not have A alleles to pass on. Let's continue with whatever else we can here. This parent, AI, why couldn't he be AA? Well, if we and this is a problem that takes a little bit of insight. He has to be AI, even though we don't even know what blood type this father is. Because if this individual was, let's say, had a B allele there, it would show up in the next generation somewhere, and we can't see that. So again, this A allele had to come from somewhere. It didn't come from this parent, so it had to come from this parent. And so that means this parent had to be donating the A allele. And so we'll talk about, in a minute why it couldn't be AA, but it has to be AI. So this parent then also has to be AI because the I allele is coming from this parent. The A allele had to be coming from this parent. So that accounts for the A allele here. So this, again, is a little bit of a tricky one. This offspring here, we don't even know what blood type the son is, so it could be AI or II. So we don't really know just because we don't even know the blood type, but it has to be one of those two given these parents. And again, you can set up Punnett squares showing these alleles and look at possible outcomes there. Okay, now, as we come back to these two parents in BI and type B and type A, this individual is going to have to be BI, and this individual could be AA or AI. So the reason this individual cannot be BB is that if this individual only had B alleles to pass on, we would see them in this son. Okay, so again, this is the codominance, and so again, this takes a little bit of insight. It's kind of complicated. So again, this individual has to be BI, because if this individual only had B alleles to pass on, this son would have to have a B in the blood type, either type B or type AB, and it's type A. Okay, and then also this individual can't be II, obviously, because they're type A here. All right, so this individual over here then we know is contributing the A allele, and so this individual could be AA or could be AI. could be either of those two alleles. And so we know the allele is coming here. We know that the B is not coming from here, that the I has to come from that. So this individual has to be AI, because again, if this was BB, it would be AB type. And the A has to come from this parent. So that's what necessitates this individual to be AI. And then that leaves us with two daughters. We don't really know what their blood types are, and so there are a lot of possibilities here in terms of genotypes. If you cross AA with BI or AI with BI, we would want to look at all the possible outcomes here. So individuals could be AI, they could be BI, they could be AI, or they could be II. So these are all possible outcomes given this cross. So the Landsteiner crosses are pretty complicated because we're throwing in another monkey wrench here. We're still looking at one gene, but we're looking at multiple alleles, several alleles in this case, 3 alleles in this case, and as a result there are multiple genotypes and multiple phenotypes compared to what we've talked about in the past. So it just complicates matters. Okay, now let's go back to a couple of simpler problems then. So again, that one's more complex. You certainly want to look at that problem again and then look at your problem sets and be sure that you can do that. But let's go ahead and look at kind of a variation of what we would see in pedigrees. So sometimes the pedigrees are described to you rather than just in a diagrammatic form. So looking at guinea pig fur, brown or black is dominant to white. And so again, white would be recessive here, and we're just talking about 2 alleles now, 1 gene. So in this kind of a situation where it's described to you, where we have a brown parent and a white parent that has 3 brown offspring and 2 white offspring, what are the possible genotypes here? So again, start with what you know. Always start with what you know. The individuals that are white then have to be homozygous recessive. And that means that this parent only has recessive alleles to pass on. This had recessive alleles from both parents. So that means that the brown parent is heterozygous rather, and the offspring are heterozygous. So again, now that problem looks really simple. But the point is that pedigrees can be either described to you in a problem or they may be out in front of you in a diagram. But where do you want to start? Even if it's something more complicated like Landsteiner blood groupings. Start with the phenotypes that tell you the genotypes and then follow the alleles either down the generations or up the generations. All right, well that's it for today and see you next time.